Welcome, everybody, to all those here in the auditorium and all those on Zoom from across the world who are joining us for this very, very special day. My name is Deepak Srivastava, and I have the privilege of serving as the president of the Gladstone Institutes in San Francisco and as the director of the Roddenberry Stem Cell Center at Gladstone. And today we're gathering to celebrate the Ogawa-Yamanaka Stem Cell Prize 2023 award winner, Magdalena Zernicka-Goetz. And this is a prize that you'll hear a little bit more detail about from Shinya Yamanaka in a moment, but one that was started by Hiro Ogawa and his wife, Betty Ogawa, and continued now with their sons, Marcus and Andrew Ogawa. And this prize was set up to recognize scientists who are doing work in the stem cell and cellular reprogramming area that could really impact human disease in the future. And it's the wonderful gift that they made some years ago that we're able to administer this prize. And we are very fortunate to have, as a co-sponsor of this award, Cell Press, and they've been wonderful partners throughout this period. We have a very distinguished selection committee made up of George Daly, Hideyuki Okano, Fiona Watt, Lawrence Studer, myself, and Shinya Yamanaka. And we received many nominations this year. And the awardee that you'll hear from in a moment was a unanimous selection by the committee for the outstanding work that you'll hear about. Now, we stand at a moment in time right now where the promise of stem cells that many of you have heard about for years is finally coming to fruition in many clinical trials all over the world. There are at least 15 different diseases now for which stem cells are being introduced into people in various trials, many of them yielding initial results that look amazingly positive, for example, for diabetes, visual loss with the corneal transplants, et cetera. And I think over the next few years, we'll see the results of those as they read out. Not all will work, but many of them will. And one of the milestone events that occurred in this field was by our own Gladstone investigator, Shinya Yamanaka, when he made the discovery that one could take cells from any adult, their skin, or now even a teaspoon of blood, and in a dish, turn those cells into something that behaved just like a human embryonic stem cell. It obviated the ethical debate around human embryonic stem cells, and it opened up a whole new avenue to understand human disease, human development, as you'll hear in a moment, and really revolutionized our field. And for that discovery, Shinya received the Nobel Prize in 2012, along with Sir John Gurdon from Cambridge University, who I'll say more about in a moment. We're very proud that all of you know at Gladstone, many of you know we have just two missions. One is to do discovery science that can impact humankind. And the second is to train the next generation of scientists. And Shinya is a great example of that, as he did his postdoctoral fellowship training here at Gladstone in the mid-1990s before going on to make his landmark discovery. So at this time, let me ask Shinya to say a few words about the Ogawas and this prize. And Shinya, as most of you know, is a senior investigator here at Gladstone and runs a laboratory here. And for many years, since 2007, while he's been here, was also directing the Institute's CIRA in Kyoto, Japan. And he has made phenomenal contributions to our both understanding of stem cell biology and its implementation into clinical trials. So, Shinya.
Good morning, everyone. Let me talk about Mr. and Mrs. Ogawa. Myself and my wife Chika make it a rule to visit their cemetery every year just before this prize. So Mr. Ogawa was a very successful businessman. He started his company when he was 55 years old, The company was about container shipping business, and he became the first Japanese who made his company public in the New York Stock Exchange market. And then he decided to give back to society. He supported many activities such as Red Cross, US-Japan Council, Asian Art Museum, and also San Francisco Opera. He also was a great supporter of science, including ISSCR and Gladstone Institute. So that's how I met him, actually in this room, like 15 years ago. We went to Stockholm all together to receive the Nobel Prize. And before his wife, Betty, passed away, they decided to create this award to celebrate breakthrough scientists in practical medical application of stem cell technology. So Hiro and also Betty, they thought differently. They wanted to do something very unique, very unusual. And I'm sure they want to recognize scientists who do the same. In that regard, this year's awardee, Magdalena, is the perfect scientist. So I really love her science. It's amazing. It's good, unusual. So thank you so much for accepting this award, Magdalena. We are all waiting for your talk. So thank you very much again.
Let me say a few more words about our awardee as I introduce her. So Magdalena grew up in Poland and went to the University of Warsaw for her undergraduate degree and also did her PhD degree there. And then as in the 1980s, as you all know, the wall came down and she had the opportunity through a fellowship in around 1990 to spend a year of her PhD at Oxford through a scholarship. And that opened her eyes to this type of science that was being done there. And she ultimately did her postdoctoral fellowship with Sir Martin Evans at Cambridge University. And there she was able to combine the work she did in her PhD work, which was really studying embryos. And she might say a little bit about that. And in Martin Evans' group, as many of you know, because he won the Nobel Prize for stem cell field as well, learned about stem cells. And then in her own lab that she started then in 1997 at Cambridge, put those two areas together that has led to this wonderful journey that she'll talk about today. And she's really been recognized for quite remarkably being able to understand mammalian development in a way that has allowed her to understand the principles of self-organization that has allowed stem cells in a dish under the right conditions to self-organize into basically embryo models. So they self-organize into things that can gastrulate and can develop initial organs in a dish just simply from the pluripotent stem cells, which is really remarkable. She's been recognized by many awards recently for this work, including the Conklin Award in Developmental Biology. And also this year was named by Nature, her discovery was named as one of the 7 biotechnology advances to be watched in 2023. And she's done all of this while really running a remarkable laboratory and group of trainees in Cambridge. And in 2019, was after rising to full professor there, was recruited to move to Caltech in Los Angeles. And she's been in transition since then and is running a laboratory in both places currently and is a professor at Caltech. And several members of her laboratory, I believe, are here or will be here later. She has also, while doing this, been married to her husband, David Glover, and they have 2 children, Natalia and Simon. her husband and son, who's in LA, contracted COVID a few weeks ago and couldn't be here today. But maybe they're getting over it. Maybe they'll show up tonight. But I think she's a wonderful example of being able to do great science, being great at all other aspects of life that are important. And she's even along the way written a book called Dance of Life that you can all buy on Amazon. A little plug for you there, describing her journey and some of the discoveries that she's made. So Magdalena, we're delighted to present you with this honor. And at this time, if you'd come to the stage along with Shinya, we'll present you your award that I'll tell you a bit about, and a $150,000 check that I see is not here, but check's in the mail. So come on. I think we're going to stand right here with some photographs, and Kelly's going to give me this. So let me tell you a bit about this crystal. When we started this prize some years ago, Shinya and others and I here worked really hard to have a local artist design this special crystal, which actually is quite heavy, as you'll see, but it has the Waddington landscape at the top that indicates the concept of cellular reprogramming. And Magdalena, we're delighted to present you with this award.
Thank you so much. Wow.
If you'll come stand here, we'll get a picture for the audience and hold this up, straight up and down like that. Yeah, great. Well, congratulations.
Thank you so much. Thank you.
And I'll take this and hold it for you.
Make sure to dance.
And now I present to you all Magdalena.
Thank you so much. And I really wish to start with the thanks to many, many people. And I will talk about many of the people's work during my talk. So I'll start with the thanks to the Gladstone Institutes, DPAC Selection Committee, and the Ogawa family for this enormous honor. I have to say that you can't believe how much it means to me to be awarded in the name of Shinya, who is my hero for so many years. And as you see, I actually met Shinya many years ago, and to be able to think about those events, this was 2014, as Shinya and I just discovered. We were not I was not 100% sure which year it was. I would never predicted that I would be standing here and speaking in Shinya's name. So I have to say thank you so much, and thank you all for coming to celebrate this event with me. So I took this photograph, as I've mentioned, in New York 8 years ago of Shinya and John Gurdon, who is one of my special mentors in Cambridge. And yes, when you think about their work, the work that they got a Nobel Prize for in 2012, it was really to show that we can take any differentiated cells from the body and bring back its journey to the state when it's pluripotent, so reprogram that cell. And when I think about this discovery, we know all that it totally revolutionized research and medicine. And when I think about it, and what I am doing is sort of opposite route I am taking. You can think about it as opposite route, because I'm taking these pluripotent stem cells and see how we can convince them to work together to create a new life, to set up a brain, a gut, a lung, a placenta, and then work together to make all of those organs in coexistence. And the way we are trying to do this in our laboratory is by building models of complex embryos from scratch. They are not real embryos, they would be embryo models, and I would try to make sure that I always refer to them as embryo models, that embryos, if one time I skip and call them embryos, no, it's wrong. I should not call them embryos, as they are embryo models. So I would like to tell you why I actually thought that we should do it actually 10 years ago. Actually, the same year, 2012 was the first time when I was thinking about this idea. So maybe this is a magical moment in my life too. So why did I wish to build this embryo model? So there are two reasons for it. First is to uncover the principles of self-organization. So I was always fascinated by the mechanism that embryos can be put together. Actually, they put together themselves. So we were trying to understand this. And second reason is that many pregnancies that I will show you in a second fail at the very early stages of life. This is actually the time when embryos self-organize. So not always the self-organization, so remarkable, happens correctly. So I wanted to understand the reasons for that failure. So when you look at every single creature that I'm showing on this slide, you'll be perhaps as impressed as me that they're all very beautiful and complex. And here are my two children. This is Natalia, older, and this is younger Simon. They're actually older, a little bit older now. And when you look at all of those creatures on the slide, you will be perhaps amazed by the complexity, particular form. They're unique. And yet, every single creature on that slide started in its life in exactly the same way. So through this process of fertilized egg cleaving into smaller and smaller cells, the cells then talking to each other to identify the way they can collaborate together to form different parts of our body. So the question is how this complexity can arise from very humbled beginnings. So the second reason for my interest is this clinical relevance. So majority of the pregnancies fail when women do not even know they're pregnant. These failures happen within the first 2 weeks of our life. So around 30% of the pregnancies are believed to be eliminated by the time embryo implants within the body of the mother, and around 30% at the time of implantation and soon after. So what it is, so what is it about these very early stages of our life that our embryos find so difficult to get right? Why their life is so fragile? So to be able to address this question, to a certain extent, we can study mouse embryo development, and this is what we have been doing for many years and still do. And this is because human and mice embryos develop in a very similar way, particularly up to the point of implantation. So for many years, I was trying to understand with my lab how it is that you start to form different type of cells. And we still try to understand the mechanism behind it and how those early cell fate guidance that happens in the very early embryo shape decisions that leads to these three lineages. So the cells in red here are called the epiblasts, and they are the cells that will give rise to every single cell of the future body, both in human and in mouse. The cells in blue are the ones that will make a yolk sac. So this is the structure in which the body of the embryo will grow. And the cells in brown will make trophectoderm. This is the structure that will create placenta. And that's the organ that combines the body of the embryo with the body of the mother to survive within the mother. So we know quite a lot about these first few days of our life. In the context of human life, it's the first 6 days, because mouse and human embryos can be cultured outside the body of the mother in a very simple media. So we can observe those embryos and manipulate them. But what happens next is extremely difficult to understand. And this is because this blastocyst stage embryo, with these three lineages formed in the right place, have to be put back to the mother for the process called implantation. Essentially, it is invasion of the body of the mother. The embryo is still very small at this stage, so it's extremely difficult to follow. And it was not possible at the time when I started my research to culture successfully even mouse embryos through this period of implantation. And of course, it was not entirely possible for human embryos at all. So to be able to gain access to this information, what happens at the time of implantation when these three major actors are now put on stage, the three tissues, but now they have to act and collaborate together to form new organisms, we had to develop two approaches. Well, I thought it would be cool to develop two approaches. The first approach I thought we should start with, and this is to take those pre-implantation mouse, as I'm showing here, but also human embryos, and culture them through implantation ex vivo to really understand how this communication between cells work. And then to use the knowledge that we will gain through the system to attempt to take stem cells for these three lineages and put them together or allow them themselves to be sensing who is who and self-organized. I have to admit that 10 years ago, I thought that approach 2 might not work. Approach number 1, I thought it was just a matter of finding the right cocktail of growth factors. It was much easier than I thought with the approach number 1, and was very hard with approach number 2, but now they are all coming together. So I would like to say first about mouse embryos a little bit, as while I've mentioned that mouse and human embryos develop in a very similar way, mouse embryos are very easy to access and human embryos are not. So when we were trying to establish conditions to culture embryos through the period of implantation, we did a lot of experiments to tune the conditions, platform and media on mouse embryos. And here is one of the very first embryos, mouse blastocyst stage embryos, that is put in that platform. And as you can see, the cells initially are sort of attaching to the medium. Attaching to the medium, to the culture dish, and then sort of come into contact with the media that we prepare for them and start to grow. And the structure changes into the cylindrical structure that is very characteristic for the mouse. So this system, establishing this culture condition, allow us for the first time make time-lapse movies of development and look at the interactions between individual cells. So there is one magical event that happened at this stage of our life. This is the stage when we break symmetry, not actually for the very first time, but one particular breaking symmetry event is very important. This is to set up our major body axis, to decide where we are going to put the future head and the rest of our body. This happens soon after implantation. And we were always interested in this symmetry breaking event. And I wanted to understand the reasons and mechanism of how embryo decide where to make this major body axis. So we used that system. This was our first question to address using this system. We knew it happens around the time of implantation, but now we can take mice embryos that mark different structures and follow development of the structure. So what is shown here on this drawing and another example of the cultured embryos in this system allow us to identify the origins of this center that is called anterior signaling center. I mentioned this name, it's complex because I would talk about it a little bit more towards the end of my talk too. And this is the center that is established in one of these two tissues that actually do not form the body. The extraembryonic tissue, the tissue will form yolk sac later, but this center will decide which part of the tissue that will make the body will become the head. So it's a signaling interactions between that center, which actually sense inhibitors, and the tissue under that will be protected for the future head formation. So we found how the centers become specified, and we were able to follow it to this very earliest event. And I wish to say it's not only us interested in the center, there were many, many groups contributing to understand the molecular underpinning of the center generation using this system. So now we have a system. The question is, can we put human embryos in that environment and would they survive? And this is what we did next. And this was another remarkable moment, at least in my life, because we were able to culture, in theory, human embryos legally until day 14. This is the boundary beyond which we cannot go. But actually, technically, we were only able to culture human embryos until this day 6, the time of implantation, and then they have to be put back into the body of the mother. So now the question was, can we progress those embryos in vitro to find how they develop? And actually, I wonder, maybe I have it, this movie. Yes, so here you see a lot of movement. This is the blastocysts that have to attach to the culture dish. And when it attaches, you will see it on the right, it starts to grow for the first time. Actually, only after embryo implantation, cells within the embryo proliferate properly and the growth of the embryo starts. So using the system, we were able to look at the formation and establishment of interactions between these 3 different tissues that are also set up in the humans, like in the mouse. And actually, most recently, we were able to identify the same signaling center, this anterior signaling center, in equivalent tissue to the primitive endoderm tissue that it has become set up in the mouse. So we have the culture system, but actually, is it good? So for us to ask this question, we thought that we should check whether we can, the embryo, human embryo in culture, can fulfill the key milestones that we expected them to fulfill. And the expectations come from the very limited number of embryos developing in vivo that were collected in 50s. This would not have been possible these days, but was possible then. And they established so-called Carnegie collection, in which around 30 embryos were analyzed at these different stages of implantation because of the pregnancy termination. So we were looking whether we can identify now, when we look at the actual proper interactions between the cells, whether we can identify similar features as were presented on these old photographs of embryos from 50s. So we see indeed segregation of embryonic and extraembryonic cells in that system. We see the differentiation of extraembryonic cells into distinct cell types. So the trophoectoderm will form different type of cells, and we see it in that system. We see this amniotic cavity formation in the center of this epiblast group of cells that will later make the body. That's very important cavity. It's the first cavity in our life. We see differentiation of the epiblast, this tissue, into amnion and epiblast disc. We see just initiation of the yolk sac formation in vitro, although it doesn't grow at that stage. And we finally see the symmetry breaking event that I've told you is so fascinating, how the cells decide which fate they would follow for the first time. So this is really for the first time when human embryos were grown from this pre to post-implantation stages. So this was a system that provided us with a lot of information, but there are caveats. And the caveat number one is that majority of the human embryos are not accessible for research. So there are very few human embryos, relatively speaking, donated to research. And second reason is that to look at the mechanisms of specification of those cells and cell-cell interactions is actually not easy when material is limited. And these are the two reasons that led us to think that maybe we can now take this information that we have learned from culturing embryos in vitro and build embryo models in vitro as well. So in this in vitro embryo models, we will be using, in our case, 3 types of tissues, the stem cells for 3 types of tissues. So for epiblast, so the ES cells are derived from the epiblast. TS cells that are less well known, but they are the stem cells derived from this layer that will make the placenta later on. And Zen cells are derived from this layer that will form the yolk sac. So what I will tell you now in my remaining time is the stepwise journey that we took that involve taking 1, 2, or 3, or making new type of stem cells and putting them together to create different models of embryo self-organization. And I will tell you examples of the findings that we were able to make using this system. Okay, so the first model was very, very simple. We actually couldn't at the beginning find conditions that will allow us to have all 3 types of stem cells happy being together. So that's for the necessity. We decided to start with just ES cells. And we embedded those ES cells, either mouse, and then we did it for human 2 years later, in the extracellular matrix. And we found that when we plate cells either as individuals or very small groups, they are able to self-organize into those structures that look very much, like embryo at that stage from our in vitro culture system. They would form these rosettes, and in the center of this rosette, lumen would open. So this system ultimately will show it's not perfect, because to have a perfect embryo-like model, we need to add those extraembryonic tissues. Nevertheless, it reveals 2 amazing principles of self-organization. So first, when we started, it was thought that this amniotic cavity forms through cell death, program cell death in the center of that epiblast. So we found this is not the case. We found that this happens through the self-organization of the cells into this rosette-like structure in which lumen will open. And I wish to show you this drawing. I hope that Ania Hupalowska, who created this drawing in my lab, who was at the time my lab manager, and now is artistic director in Gene and Tech, she wanted to come to be with us today. She couldn't because she also got infected by COVID. But she had a lot of sense of humor and helped me with my lectures at the time when I was giving, when she was with me, by drawing the analogies that I like. And some of them I still have in my office framed on the walls. So we found essentially that, yes, that on the next image you can see that this is the polarization of the cells into the rosette-like structure, and then exocytosis of podocalaxin that leads to the opening of this lumen. So this was very good because we thought that lumen forms differently. So now we know that lumen forms through this first self-organization of group of unpolarized cells to polarized cells that form epithelium that open the lumen. The second important finding was that this process of self-organization requires exit from one particular state of pluripotency called naive state of pluripotency. So those organized cells show this naive pluripotency state by expression of NANOG. And when the cells become polarized, they still express of NANOG, but when they initiate process of cavity formation, they have to let NANOG go. So this is the transition in sulfate pluripotency. But this is not a perfect model, of course, because it only models fragment of the embryo, this fragment that make a body. Nevertheless, we know that this fragment needs these other two tissues to interact with it. And essentially, these are derived tissues from this one that will make the yolk sac, shown here in green, and this blue tissue is derived from trophectoderm. These are the tissues that signal. So actually, there are major players in development of our body. Epiblast is like a group of pluripotent cells that really wait for those signals to direct them what to do. So we have to provide those signals. So yes, and actually, particularly, we want to provide those signals and see whether we can recapitulate this event of symmetry breaking, the specification of this anterior signaling center that will decide where the future head, tail, left and right side of the body will form. Yes, so can we model this event with stem cells? So the first thing that we did is to put together model 2 by bringing company for embryonic stem cells in the form of bringing the trophoblast stem cells that are derived from the tissue that will make a placenta. We again embedded those cells in extracellular matrix, and after 3 days, we saw that they form embryo-like structures that actually extremely similarly appear with a very similar morphology to real embryos. They didn't have this layer of cells in which this anterior signaling center become established, but apart from that, they form cavity. They really look pretty handsome. So now the question is, without that anterior symmetry organizer that breaks symmetry, can those embryo-like structure break symmetry? And the answer was yes. So this was entirely unexpected to me, that when we use a particular type of stem cells that will monitor symmetry breaking events by changing expression of some of the genes, essentially inducing multidrome formation, and we monitor this by looking at the expression of brachyury. So this is GFP expressed from brachyury promoter. You see that on one side of the epiblast, we start to form the mesoderm. So this was very surprising to me, because we thought that we need this center that is missing to have it done, but clearly it's not the case. So what I think is happening here, and this is more hypothesis that we know for sure, that while in natural development we have the center migrating from distal tip towards one side to send inhibitors to specify a gradient of morphogen, so that the BMP nodal and twin signaling is very high on one side of the embryo only to form the primitive streak, to form gastrulation site. Here, when we don't have it, the local interactions between different signaling properties that become established are leading to formation of this self-patterning organization event. Actually, most likely following the rules that many years ago Alan Turing described first. Okay, so you can guess that the next model was aiming to put all 3 types of stem cells together. And so indeed, this is what we did next. And for this, we actually changed the design. We don't use any more extracellular matrix. We use this dish that put all of the cells together at the bottom of it. And so we add here ES cells, TS cells, and Zen cells. And we put them into this dish and let them develop in culture with time. So after 24 hours, those cells sort from each other, and then they start to organize into structures. Not all of them, and I will come back to it, why not all of them, but many of them will be absolutely perfect. So here you see this ETX structure, so ESTS and Zen cells together, and here you see on the right, natural embryo. So you see that all three compartments are set up in that system in the right, of the right size, of right morphology. So how far those embryo-like structures can develop? Well, they can start the process of gastrulation. They make this anterior signaling center. So you can see here, they express right genes in the right place on one side of the embryo, and mesoderm now forms on the other side. And these mesoderm cells start to leave the epiblast and form this germ layer by breaking the basal lamina that was normally inhibiting their movement. But as I've mentioned, not all of the structures look so brilliant. And actually, I believe this is the strength. It's a weakness as well as the strength of this model, because now we can ask the question, when this self-organization works, what are the major conditions that we have to fulfill, or the cells have to bring with themselves for the process to work, and when it does not. So we can decipher the requirements for self-organization by studying all of those combination of cells that work and do not. So, for example, so what we do, we can label those cells with different colors, film the process of self-organization, and see when it leads in, when we don't perturb the system, around 50%, depends on the hands as well, and on the day. As always in life, everything depends on the day, but if sun is shining, around more than 15% of structure look beautiful, but if it's cloudy, then around 50% of the structure looks like that, and 85% have some abnormalities. Some cells will go to the wrong compartment. So by filming development, but also by doing a single-cell RNA-seq on those cells at different stages of their self-assembly before and after, and by doing gene modifications by RNAi, doing screens, we were able to identify this cadherin code that the cells use to sort from each other. So individual cells, Zen, ES, and TS cells, and also real cells in the embryo that correspond to those stem cells, express unique set of cadherins. And that's how they are able to know who is who and how they should put themselves together. So knowing that, we were able to modify this cadherin code by overexpression of specific cadherin, specific type of cells. And this led us to massively increase the efficiency of self-organization. So this was another good day of my life, I have to say, to not just only, we don't have a full story yet, we don't know all of the requirements, but we know that this cadherin code is very important. And when we culture cells in 2 dimensions, the cell become heterogeneous and can start to express cadherin at different levels. We also know that cortical tension, regulation of cortical tension is very important. Okay. So this is like a short summary before I will tell you a few things more, but I wanted to say is that really this is by uncovering this process of natural development, particularly at the time of implantation and this early post-implantation stages, but looking at embryos continuously, mouse and human, we're able to build these models that let us create this complete embryo-like structures. And I wish to acknowledge here another lab manager that I had the pleasure to have at the time, Andy Cox, who actually drove those. When Anya left, actually Anya stayed in the lab, but she became a Marie Curie fellow in the lab and started to do more scientific project and manage our lab. Andy Cox took a role of lab manager, and he was also a senior postdoc at the time in the lab, and discovered that he has this talent as Anya, or similar in context of the embryo development images, and did draw all of those images that you admire now. So we found that the structure actually have a potential to develop even further than to gastrulation. But for that, we have to change not ES cells, they are perfect, but we have to replace Zen cells with ES cells in which we induce expression of transcription factor that is very important for formation of this lineage, this primitive endoderm lineage, GATA4. And we also can now replace TS cells by inducing expression of transcription factor CDX2 in ES cells. So now when we put all of the cells types together, they actually can develop all the way until early organogenesis. So and I'll show you an example of head-like structure and height-like structure that they form. And so you see here some real artist images of how those embryos look like. So actually, this is the image that we propose for a cover. Nature didn't choose it, but this was one of the first authors of that work that did this image. That's why I wish to promote it. And here, Cell Stem Cell choose the image. We are very happy done by professional artists of how Casey, in this case, hens, are creating those structures. And I will show all of the heroes of those studies at the very end. Normally, I have a picture of every blood member in the right corner, but here, this time, I decided to put them all together at the end to acknowledge their work. So here's an example of the organs they make. So this is the brain-like region, very similar to brain of the natural embryos, as far as we can see with the markers, and we still study it in more detail in feather and also push the system feather. This is heart. Like structure that is beating, so we think it's functional, and it develops on day 7 as to the same extent as 8.5-day-old heart in the natural embryo. This cultured in vitro in our hands. So those stem cell-like models of embryos, they also specify cells for the next generation. They form primordial germ cells. And the right compartment, so they will be specified the boundary between embryonic and extraembryonic compartment, and they are specified in similar numbers, and they grow as one would expect them to grow in vivo. So this was all mouse for now, apart from in vitro culture system. So the big goal is now, this was the big question over the last several years, can we do the same with humans? So this model is much more important, human embryo model, because studying human embryos is very, very difficult if you are interested in the mechanisms. You can observe them now because we have this in vitro culture system, but we cannot generate transgenic human embryos. So can we create similar model for the human embryo? So this was really my dream for 10 years. And it's only this year that I can show you the data that convinced me that the answer is yes, we can. They are not yet perfect, but we can create the models of embryo, human embryo development at the time of implantation. So in our lab, we're particularly interested in trying to mimic these peri-implantation stages of development leading to gastrulation events. So the first task which we had to overcome is to identify which transcription factors we should induce to be expressed in cells. And we did want to do it through induction because the addresses, the transcription factors the programs of those cells would be different from one cell for trophoblast-like lineage and hypoblast-like lineage. And yet we want to put them into the same media. So we thought that looking at the addition of growth factors to the media might be confusing for these cells. So what we did here is we look at we sort of score transcription factor activity across the lineages and settle two different transcription factors, GATA6 and SOC17 for hypoblast and GATA3 and epi2 gamma for trophoblast. So then we confirm first in 2 dimensions that indeed when we induce expression of those factors, and we do it in different pluripotent cells, different type of pluripotent human stem cells. That's why we have 3 rows here. But we look whether they are able, these transcription factors that we chosen, indeed start expression of other factors that are important for this program of those lineages, and the answer was yes. So then we put those cells together, again using the same dish, and using essentially copying our approach that we developed for the first time in the mouse years ago. And after 4 days, we saw these self-organized structures. Now, they look pretty miserable, and when they're in this dish, you don't know whether they're good, so you have to take them out and characterize them. And that's what we did. And this was incredible. Again, another day that I wish I would never forget. We saw that those structures organized into the central domain resembled as epiblast, and indeed was derived from the ES cells. And outside were hypoblast-like cells and trophoblast-like cells. So we characterized those structures in much more detail. So we saw that the structure opened the cavity again through this polarized secretion of podacalacin, and the efficiency of the system is around 25%. So efficiency of luminogenesis in the structures is 25% on average. We characterize expression of many different markers that I'm showing here for different lineages to really understand what happens. Now, I have only a few data slides left to show you, but I would like to show you some findings that we have made with those structures. So the first thing that we wanted to do is to understand how good or how bad those structures are. So we know that luminogenesis happen very, very well, but what about the further trajectory in life? So for this, we did gene expression profiling and chromatin architecture profiling to understand structures at day 4, 6, and 8. And we're able to see that epiblast develop very well into the amnion lineage and also go through different steps of differentiation towards mesoderm intermediate lineage specification until extracellular mesenchyme. Hypoblast also developed very well, but trophoectoderm initially developed well, but then it started to also express endoderm markers. So there is one minus in that system, that it looks great, but trophoectoderm at one point is losing its fate. Nevertheless, all three types of stem cells were absolutely essential for form those structures. If we remove one of them, the structures would not form. So we wanted to know how far those structures can go, and even if they are not perfect human embryo structures, what they can teach us. So one amazing discovery that we were able to make using those structures is observation of human primordial germ cells formation. So actually, we can identify those cells on amnion trajectories. When we look at this, they become specified for the first time in day 4. Embryo, then more of them become specified in day 6. And this is so robust that we were able to carry out functional studies and block different signaling pathways and ask which of them is absolutely essential for formation of PGCs. And this led us to identify that BMP is critical for that. So if we eliminate BMP by using inhibitors, PGCs would not form. So two final slides. I wish to say those structures can actually progress further in development, and some of them do look like real embryos at day 14. So this is the limit to which we can culture natural human embryos. And here's an example of such a structure that exhibit whole hallmarks of day 14 embryo that initiate the process of gastrulation. So here we see these breaking symmetry events. So the epiblast is split into these two different parts. The scales in green are called epiblast disc, in purple amnion, and some cells, so there is one symmetry breaking event, and some cells within the epiblast start to express mesoderm markers, brachyury. So the efficiency of formation of the structure is very low, around 1%. And actually, this is very similar efficiency. I am so pleased because many people are now building those human embryo models. And several of those papers just came up this summer. And actually, efficiency of formation of this gastrulating embryo-like structure is very similar. So this is we, as a field, have to improve and see what it is that we need robustly to do to our system to behave in a better way. Although I wish to stress that all of this is through self-organization. We do not stimulate any specific thing, any specific signaling at any journey of those cells with each other. Yes, okay, so this is my final result. It's a little bit complex, but I'll try to explain it. I hope I very rarely talk about it, but I would like to take this opportunity. So here you see this beautiful embryo in which the epiblast is splitting into amnion and epiblast disc on the other side. But many of the structures that you create, actually a majority of the structures, do not have those two splits, but all of the cells will be green. So all of the cells will be, purple. All of the cells will form amnion. So we were wondering why it is, that we do not have that split. And we know that in natural development, we need this anterior signaling center on one part of the embryo to protect epiblast from differentiation. So we look at the sequencing data, and we found that indeed in our model, we do not form this anterior signaling center very robustly. So then we went to our human embryo sequencing data, and we found that in human embryos, SOX17, that we are overexpressing in our system, predicted activity of this transcription factor anti-correlates with expression of the factors that make this center. So we decided to fix it by generating new line in which we do not overexpress only GATA6 and SOX17, but only GATA6. And in this way, to cut a long story short, we fixed that problem. And now this is my summary slide. We now can create more robustly the embryos that initiate the process of mesoderm formation because we do provide this anterior signaling center. There is still a long journey, I have to say, to make this model as beautiful as human embryos are. And then the question is how far we can culture it and what we can discover. But the days are extremely exciting, and really, I think that there is a lot to do ahead. But we already have that system that I am so pleased. So on my next slide, I would like to summarize this very briefly. So when I started my career, we were not able to culture human embryos beyond this day 6. It was also very difficult to culture mouse embryos beyond through the implantation particularly. So we could culture pre, we could culture post, but not through the stage. So I'm so pleased, and I wish to award my group for it, for staying on that journey with me to establish the system that now we can go through and beyond implantation with human embryos ex vivo. So we now can also build human and mouse embryo models. Mouse embryo models, I've told you, can gastrulate and develop all the way until early organogenesis in vitro. I imagine that they could go further, but this is the limit to which we can successfully culture even natural mouse embryos in our lab when we start at these early stages. Human models, they have to still develop through gastrulation better, but they're already teaching us a lot. So here are a few lessons. So first, extraembryonic stem cells are absolutely essential to direct morphogenesis. This is what this model said, and this is because we were able to put first one, then second, and then all three together. They are critical for correct morphogenesis. The another interesting thing about this model is that we can bypass the first few days of our embryo life. So we can bypass formation of the blastocyst stage, because when we put the cells together, they don't form blastocysts. They go immediately and form post-implantation structure. So this suggests that formation, actually the first few days of our life, is more about formation of different type of cells rather than proper organization of the cells, or at least we can bypass it. I believe that the variation in our system is strength. My lab might disagree. But I see it as a strength because although it's annoying us, we actually can leverage it to really dissect what it is absolutely that is essential to fulfill the proper development. And I think the last point is perhaps the most important point. These findings point to this inherent ability of different stem cell types to self-assemble into the biologically meaningful structure, which I think is just really mind-blowing, because we are not engineering those cells in the right places to do the job. They really do it themselves. So I think it's incredible. And because it's such an incredible point, I want to tell you something that I actually found yesterday. I mean, I knew a little bit about it earlier, but I was wanting to look at this more to present this idea to you. So very famous philosopher, Immanuel Kant, you see, in 18th century, defined life as an emergence of function by a self-organized, self-producing process. And I think that this discovery that stem cells representing the 3 first lineages naturally assemble into embryo-like structure attests to his insight. They organize and produce the parts which they ultimately need to rely upon. And that's sort of how I now think we can think about our body as a symbiosis of several parts that allow new life to exist. It's a concept we can discuss, a more philosophical concept at the end, but I would like to present this idea. So the important thing is that now we can begin to analyze development from this perspective and determine the molecular and physical basis of how self-organization emerges, to be able to identify those principles that some of them we identify, but not all of them yet. I wish to finish with how I started, because now we are trying to use Shinya's iPS cells to create those models for human. So stay tuned. Hopefully we can succeed with this. And at the end, I would like to show you two most important slides. So first, with all of my current lab members, some of them are with me here. This is incredible group of people that I have a privilege to be part of. And I am so grateful to them that they stayed. The journey not always was the easiest, because when we started, we didn't know it would work. So I am very pleased that it did. Otherwise, perhaps, well, I would not be here, but also we would have harder life. I would like to thank them. And I would like to thank all amazing collaborators that helped us and make our labs fun. Funding bodies that trusted in the progression of this research at a certain stage, that it will result in some results that we can, we can contribute to. And finally, I would like to show alumni from our lab that now they have their own labs, majority of them, or they have another important roles in science. All of them stayed in science, although Carlos is on a job market, so I hope he will stay in science too. And these are the ex-lab members that contributed to this research that I was talking today about. So thank you very much for listening. And it was a pleasure. One more time, I wish to thank and how humble I feel and grateful I feel for that award.
Magdalena. I hope you all can appreciate the import of this work. It really gets to the essence of how life starts in us. I hope you also can appreciate that such discoveries take persistence and commitment over long periods of time to really get to the answers, and the journey is yet to unfold. We do have time for some questions from the audience or our Zoom audience.
I have a question for you. So some of the decisions that we are making in terms of early development, we are always discussing about transcriptional decisions, activation of genes. And yet there are some changes that will require a much faster turnover or timing. And they probably rely on interactions of proteins and changes in post-translational modifications. How do you think about this, and how do you think your model would be able to explore those unanswered, or like the black box of the understanding?
Thank you. That's a very good point. It's true that in this talk, I only showed single-cell RNA sequencing data when I was talking about the individual players, or I was playing with transcription factors. But you are absolutely right. Many of the things happen on the protein level, and modification of to protein is very, very important. We started to look at, so there is now possibility of using single-cell proteomics to identify the changes in the protein that we use to look at the very early stages of mouse and human embryo development, where many proteins actually are critical because they are maternally deposited in the oocyte. So yeah, I'm aware of this, and I'm so happy that you are bringing it, because looking at the proteins would be very important. And of course, they are the action players. And we started to look at this special now proteomics in the context of stem cell derived embryos as well, but results are not yet ready to be presented. Thank you.
Any questions, Bruce?
The critical step of forming the cavity, I always sort of think of it as like a balloon blowing up, as it were. But if that were the case, there would be pressure that would be created in the center. And I was wondering, is there any evidence that there's like a, like basically an ion pump up, some sort of channel which is creating that pressure to open it up, or is my hypothesis just completely ridiculous?
No, it's not ridiculous. Yeah, so you are absolutely right. And I think the pressure of that cavity is actually going to, I don't have much data to say, but I agree with you. And I believe that pressure within that cavity would be very important to regulate embryo size. So this is something that has not yet been looked at properly. But I believe that's what this cavity, one of the functions of this cavity is. Another function, I think, of the cavity, your question gives me opportunity to say something I was planning to say, but when I was talking about the amniotic cavity formation, I think that it's actually, it will be a little bit philosophical what I say, but I think that there is some truth in that philosophy, that when you look at the self-organization at these initial stages, one thing that the cells do, they have to open that cavity. And actually, tumors do not open the cavity, as far as I know. I don't study tumors so well, but from what I know, this opening cavity is very common for early stages of embryogenesis or formation of the organ as well. So what's the function of the cavity? And one possibility would be that it is a sink. But I actually think that all of the cells around the cavity, the fact that that there are a lot of things secreted to the cavity, allow those cells to communicate with each other and synchronize their behavior. Actually, this is the also important function of that cavity, but I don't have any evidence for it. It's more philosophical than based in science right now. But that's how I think why we need to make that cavity first to start everything.
So I was fascinated by your talk. Again, thank you very much. So as you showed, mouse and human embryos are very similar prior to implantation, but they become very different after implantation. So I always wonder why that is. Now that you can recapitulate those by using stem cells, do you think you can answer that question in the near future by mixing human ES cells and mouse TS and Zen cells like that?
Excellent question. I haven't done that yet, but I agree with you. That's the question that is now possible to address. And if sometimes when we mix cells together of two different species, the one cell type outcompetes the another. That's the actual technical difficulty that we experience in the lab. But we have a license for doing that, because this actually requires making mouse-human chimeras, even at the very early stages of development in the context of the model, is guarded, but we have a license to do that to exactly address this question. How can we go from this eccentrical structure to the flattened disc? And which of those three types of stem cells is taking the lead here to shape the morphology of the structure? Excellent question. Thank you for asking. I hope I'll be able to address it.
Magdalena, when your paper came out with the human embryo models, it got a lot of press, and there was also a lot of discussion about ethics around that. I wonder if you'd comment, because we haven't really talked about that yet, the ethical structure around which you're doing your work related to creation of human embryos.
Yes, so thank you for asking this, Deepak. Indeed, I first wish to say something. That when I presented this data at ICCR meeting, actually it was not first time I presented this human embryo model. I presented year earlier on the Conklin Medal Award lecture. Nobody picked up on that, but many journalists must have happy to come to ICCR meetings, and that's when it appeared in news even before the paper was out. And the journal, the paper was coming out in important journal. They didn't allow me to talk about the details of the structure. So this created a lot of excitement on many fronts. But it gave also the opportunity to talk about ethics and legal issues with the study. So as we know, studies with human embryos are very strictly regulated to this day 14. In UK, actually in California, we can culture human embryos until day 12, and it's different thing that what we do right now with stem cells, because we create human embryo models, not human embryos as such. They do not have potential to develop into the human beings. They are this early stage of development of those structures, so they do not themselves at that stage of our life. We cannot equate them with a threat of whether they one day might be able to help us to change our reproductive journey. I don't think so. But what I wish to say is that it's very important to regulate this work as well. So all stem cell work is regulated actually in our laboratory. So both in Cambridge and Caltech, there are strict rules which cell lines we can use, which we cannot use, how we can use them, what we would like to do. And I think this happens, I believe, in all of the scientific laboratories that create these models. The question is now how far these human embryo models should be allowed to be cultured. And there is the discussion right now ongoing. Also, should we terminate culture of human embryos at day 14 if they can teach us about the process of gastrulation if we were to extend it for extra 7 days? It's another topic of the important discussion that is ongoing right now.
Great. Thank you very much. And please join me in congratulating once again our 2022